Welcome to the 2022-2023 Grad Slam here at CSUB. My name is Dr. Denver Fowler, and I serve as the Associate Dean of Graduate and Undergraduate Studies, as well as the Director of the Graduate Student Center. I'm excited about today. It is my distinct honor and privilege to welcome you to the competition. Before we start, I do want to thank some individuals that have been a big part of making this happen. First and foremost, I want to thank our workshop presenters, Dr. Vega, Kelly O'Brien, as well as the Graduate Student Center staff, Martha Manriquez, Adrianne Silva, Rudy Lamelli, and Emma Barnes. Also want to thank our application review committee, Dr. Zinko, Dr. Dell, Amico, Dr. Jackson, and Dr. K. In addition, I just want to preface our competition with quickly reviewing what is this competition. So what is Grad Slam? So essentially all of our participants today are going to give a 3-minute thesis, think elevator speech, focused on their research. And it's going to be given in a TED Talk style presentation, although we are doing it virtually this year. And they have one slide to utilize during their presentation. And again, they can't go over 3 minutes. I also want to congratulate all 11 of our participants. This is a big commitment, and I want to just share each of their presentation titles there and their faculty advisors, and then I'll reintroduce them as we're going through the competition. But this was a big step for all of these students, and it's also a big challenge to try to present your research in 3 minutes when you could spend hours talking about your research. I also want to recognize our guest judges. Dr. Deborah Jackson serves as the Associate Vice President for Academic Affairs and Dean of Academic Programs here at California State University, Bakersfield. And she's a longtime Grad Slam guru, having ran many of the past CSUB Grad Slams, as well as the CSU 23 campus-wide Grad Slam competition. I also want to thank Terry Church for serving as a judge. Terry has been with Dignity Health since 2008, serving as a Vice President, Chief Nursing Officer, as well as a Chief Nursing Officer for Dignity Health Central California Division. Terry has helped oversee an expansion of services, including oncology, stroke, and catastrophic burn care. She is a passionate advocate for nursing education and thoughtfully balanced the care of patients with the emotional toll it has taken on nurses during the COVID-19 pandemic. Notably, she's a CSUB alum, Hall of Fame inductee, and has been recognized for her achievements in the nursing field. Terry joined Memorial Hospital in 2008 after 26 years at Adventist Health and 2 years as Director of Nursing at Comprehensive Blood and Cancer Center. And our last judge, J.C. Fickle. J.C. was named an out-student graduate student for the School of Natural Sciences, Mathematics, and Engineering here at CSUB in 2021. She won the Distinguished Master's Thesis Award in STEM disciplines from the Western Association of Graduate Schools. Her thesis research examined stem and root structure and function of woody plants in the Sierra Nevada mountains. She is currently completing her PhD in ecology and evolution at the University of Utah, and her research is focused on forest health and the changing climate. Please join me in giving our judges a round of applause. I just want to preface this presentation with, we had one participant who had a scheduling conflict. To make it fair, Briana recorded her presentation. It was one take. There were 2 individuals, part of the Graduate Student Center staff, who attended her presentation. So just like everyone else today, she had one shot at it. I want to introduce Briana Acevedo, and her title of her research is Determining the Origin and Formation of NWA 11788, a new lunar meteorite. Her program is Geological Sciences, and her advisor is Dr. Katie O'Sullivan.
So how do we get lunar meteorites? In order to understand this, we need to go back to the early history of the moon, when there was a lot of impact bombardment due to materials from space. Now, these materials ended up impacting and ejecting pieces of the moon off of the surface, and eventually those pieces ended up landing on Earth. And so these locations that they're usually found on are in areas like Antarctica and Northwest Africa. And Northwest Africa just happens to be where our meteorite was found. You're probably wondering, well, don't we already have samples that we brought back? So why do lunar meteorites matter? Well, studying lunar meteorites can give us information on locations that were not sampled by Apollo and Luna missions. This is because since the bombardment ended up excavating pieces of the moon deeper from within, the lunar meteorites have that advantage versus Apollo samples that just collected from the surface. And so the focus of my research these last few years has been analyzing the sample using different techniques. Such techniques as microscope observations. And so this image here on the right is actually a sample of our meteorite as a thin section. And what a thin section is basically we cut a piece of the meteorite off, we glue it to glass, and we sand it down very thinly so that light can pass through it and we can see the minerals within it. We also use other types of geochemistry techniques such as electron microprobe work and laser ablation mass spectrometry. And what these do is they basically tell us the geochemistry of our sample. And the geochemistry can then tell us information about the chemical composition. And with that, we can learn things such as possible launch sites of our meteorite, and which again, launch sites just means where it was ejected from the moon, and as well as the crystallization history of our sample. And so using these methods can help us understand more about the moon since it gives a more accurate representation of the interior. And so the main goal of my research is to basically add to the growing literature on return mission samples and meteorites, because hopefully they can fill in gaps missing from other analyzed samples, especially since we now know that return mission samples only represent about 5% of the moon's surface. And since one of NASA's top priorities is to eventually return to the moon, understanding as much as we can about these different compositions can help them prioritize locations for sample collection in the future. Thank you.
Let's give Briana a round of applause. Thank you, judges. Our next presenter is Emma Barnes, and the title of her research is To the Annoyance of His Fellow Patients, Mental Health in 19th Century America. Her program is History, and her advisor is Dr. Kate Mulry.
What's the first thing you notice about someone? Mr. G was a unique man who lived in New York in the late 19th century. As you can see from the slide, he was a talented artist who sometimes struggled finishing projects. He wore eccentric capes and hats, and he happily sang to the annoyance of those around him. It was because of these behaviors, rather than his mind, the doctor at the asylum he lived at diagnosed him with paranoia. At this time, industrialization, the idea that bigger is better, drove reform for mental health hospitals, prioritizing the quantity of patients far over the quality of care. People were questioning religion, and the new discipline of psychology opened mental illness to academic discussion. This evolving society is where G broke social norms and was categorized as mentally ill. Other scholars have not examined these behavior-based categories of mental illness. I want to stress here that I am interested in social and physical evidence of mental illness. The term mental illness in this context is a 19th century definition, not reflective of the modern meaning, though it is where our definition originates from. I began with evidence of G's diagnosis from his doctor, William Noyes. G's style of dress contrasted an orderly society. Noyes described his artwork as unfinished, evidence he couldn't commit to work. He sang regularly, which Noyes interpreted as evidence of illness because it annoyed others. All of these evidences focus on others' perceptions of Mr. G. Noyes also points to G's skull shape as physical evidence of his mental illness. At this time, pseudosciences that were precursors to eugenics were used to create hierarchies of race, gender, and class. Noyes linked skull structure to G's behavior, suggesting an additional social hierarchy. Those at the bottom were mentally ill. When Noyes diagnosed G with paranoia, he based his interpretation on observed behaviors and skull shape, not the inner machinations of G's mind. Mr. G broke social norms. As a result, he was labeled mentally ill. Those who did not fit as cogs into the American industrial machine were ostracized and often labeled mentally ill, falling to the bottom of the social hierarchy. This system is the basis for our modern institution. So I ask again, what is the first thing you notice about someone? Thank you.
Great job, Emma. Let's give her a round of applause. I would like to ask Emma, could you please share with us how you became interested in your research topic?
Yeah, so a couple of years ago when I started at CSUB, I took a class called Science, Medicine, and Empire with Dr. Mulry, and it was there that I really became interested in medical and scientific history. And this semester is my first semester in the grad program, and I am taking Dr. Mulry's course again, one of her graduate courses this semester. And in the archives on our first day of class, she mentioned a project on mental illness might be interesting. And I've struggled with mental illness, as everyone probably has, but I've struggled in the past as well. And I really thought that it would be interesting to look at it from a historical perspective. And I ended up finding this journal article on Mr. G that this man, William Noyes, wrote in 1888 that really just stood out to me and was really interesting. And I decided to do a deep dive into this.
Awesome. And if you could, just please share with us, how would you hope to contribute to the extent research and literature on your research topic?
So the evidence of illness that I'm looking at, which is really a lot of social and a little bit of physical, it seems to be kind of a stepping stone in a lot of the secondary sources that I'm reading. So a lot of historians have acknowledged that it exists, but they haven't really explored it. So I really want to sort of develop that stepping stone. That way, it kind of serves as just help in a lot of other historians' work they've already done. I know it sounds kind of backwards, but it's not denied that these evidences exist. It's just not something that's been explored. So I just want to sort of help other historians and explore an avenue that hasn't seemingly had the attention it might deserve.
Thank you. Next up, we have Danica Bergin. The title of her research is Imitation is the Sincerest Form of Flattery, North American Medicinal Leech and Eastern Newt. Her program is biology, and her advisor is Dr. Amber Stokes.
Mimicry takes place in many different forms in nature and can be used to trick other organisms. Two major types of mimicry are related to toxins, Batesian and Müllerian mimicry. In Batesian mimicry, a non-toxic or non-venomous animal mimics a toxic or venomous animal. We see this in the harmless milk snake that mimics the venomous coral snake. Müllerian mimicry is where a toxic or venomous animal mimics another toxic or venomous animal. We see this in the toxic viceroy butterfly that mimics the toxic monarch butterfly. A lesser known example of mimicry is the North American medicinal leech, Macrobdella decora, and the eastern newt, Notophthalmus viridescens. Both species coexist in various areas on the eastern side of the United States. Looking at these pictures side by side, we can see that the North American medicinal leech is mimicking the eastern newt, at least in appearance. So, why would it be advantageous to mimic a newt? Well, several genera of newts, as well as some other animals, have a potent neurotoxin called tetrodotoxin, or TTX. TTX inhibits action potentials, leading to respiratory paralysis and then death. The most toxic newt, if ingested, has enough TTX to kill up to 56 people. The eastern newt is one of the species found to possess TTX. So, I wanted to find out if these leeches have TTX to determine if this is a case of Batesian or Müllerian mimicry. I used a competitive inhibition enzymatic immunoassay to detect and measure TTX levels in 9 leeches from upstate New York. With this method, we use antibodies and a color-changing substrate that bind to the TTX in the sample. The amount of color change that is measured with a spectrophotometer allows us to calculate TTX levels. Of the 9 leeches, TTX levels ranged from 0 to 61 nanograms. As a comparison point, eastern newts have TTX ranging from 5,000 to 57,000 nanograms per whole adult newt. 4 of the leeches had no TTX, but I expect that excess liquid included in sample processing for practical purposes over-diluted the samples. Future work will correct for this. Results from our experiment support the hypothesis that North American medicinal leeches have TTX, and this is a case of Müllerian mimicry. The next step in our research is to send out our samples to verify TTX presence using a more sensitive method. Thank you.
Great job, Danica. Let's give her a round of applause. Danica, first and foremost, I'm sure like most folks in the audience, you can't believe a little thing like that can kill 56 people. Amazing. Please share with us how you became interested in your research topic.
I had been working with Dr. Stokes on a previous project looking at TTX, quantifying levels in newts. So I had become familiar with the method already. And then when I heard about these leeches that Dr. Stokes had, and I just thought, looking at the pictures, the mimicry is just super cool. And I really wanted to find out if they had TTX.
Wait, are we saying Dr. Stokes keeps these things on campus?
No, not
That's
anymore.
Okay. And then if you could just share with us, how do you hope to contribute to the extent research and literature on your research topic?
So there's a lot of literature out there right now on TTX, mostly having to do with newts, and pufferfish also have TTX. So there's a lot out there, but what's not out there right now is that these leeches have TTX. So that has not been published at all. So we're hoping to get that published.
And I'm going to introduce our next participant, Money Ghimire. And his title is Chronic Remote Ischemic Preconditioning, Attenuates Arterial Stiffness and Changes Hemodynamic Measures in Pre-Hypertensive Individuals. His program is kinesiology, and his advisor is Dr. Kim.
The simple process of putting cuff on and off can reduce blood pressure and arterial stiffness? Let's find out. So for this study, we are using remote ischemic preconditioning, or referred as RIPC, and it simply means blood flow restoration, restriction followed by restoration for a certain time. And generally in RIPC studies, we have seen 3 to 4 cycles of 5-minute restriction followed by 5-minute restoration at 200 to 220 millimeters of mercury. Here specifically, where our participants are prehypertensive in blood pressure range, and this is important because people who are prehypertensive in early stages are linked with hypertension in later stages, and which can cause cardiovascular diseases such as stroke, heart attack, and atherosclerosis. And particularly for this study, our main measures are blood pressure changes and our arterial stiffness changes. Arterial stiffness is the index of rigidness of the arteries, and it is also strongly linked with cardiovascular diseases development as well as its related mortality. So our main question here is whether chronic RIPC reduces arterial stiffness and blood pressure after 4 weeks. So on the picture on the right, we're using automated cuff on the upper arm to induce RIPC. So for specific to the study, we are using 4 cycles of 5-minute restriction followed by 5-minute restoration at 200 millimeters mercury of pressure, and for a total of 4 weeks. So picture in the middle shows how we measure our arterial stiffness. We measured arterial stiffness using carotid femoral pulse wave velocity measure, which measures the travel time between the carotid and the femoral side at the thigh, and which provides us with the velocity, and higher velocity indicates higher stiffness or higher rigid vessels. We also measured pulse wave augmentation, which provided us with the wave reflection, and higher wave reflection is indication of higher stiff arteries and vice versa with low wave reflection. For our results, we found that for our blood pressure trend, all of our blood pressure measures were decreasing at baseline compared to 4 weeks of RIPC, and similar trend were also seen in pulse wave velocity measures as well as pulse wave augmentation measures at baseline were compared to 4 weeks of RIPC. So finally, in conclusion, this trend could potentially support that RIPC can be used as an alternative intervention to control blood pressure as well as reduce arterial stiffness. However, we would need more data to confirm those trends. Thank you.
Money, great job. Let's give him a round of applause. Could you share with us why you became interested in your research topic?
I've always wanted to work with performance enhancement or improving any kind of performance. And I came across with Dr. Kim's project, and that's how I got involved, because this is relating to the cardiovascular function improvement following alternative intervention. Not necessarily, ideally, you have to exercise to improve performance. We can use some alternative interventions as well.
Please share with us how you hope that your research will contribute to the extent literature and research on your research topic.
Yes, so currently RIPC is used usually in the acute phase. So our study is chronic. So there's few studies studying chronic effects of RIPC on other measures. And arterial stiffness measured by the pulse width velocity has never been measured before. So this could potentially, majority, our plan is to apply this in clinical settings where people have low mobility. But however, this can be applied to anyone who wants to improve their blood pressure as well as their arterial stiffness.
Again, great job. And we will move on to the next participant, Mario Gaytan. And the title of his research is Using Habitat Restoration Treatment in an Effort to Manage and Save an Imperiled Species. His program is biology, and his advisor is Dr. Lucas Hall.
When restoring habitat, habitat restoration is becoming increasingly more important, as over the past 100 years, we've lost species at a rate of 1,000 times higher than any other comparable period. Without proper habitat management, the natural ecological services that humans and other species rely on could break down. When restoring habitat, we should ensure that it is done effectively, as improper management could result in the loss of a species. My objective is to determine effective habitat restoration treatments within a degraded sand dune system with the use of the San Joaquin Valley giant flower-loving fly. I'd like to note that I haven't started collecting my insect data due to seasonal inactivities of my target species. For my research, I'm looking at inland dune systems within the Central Valley. They once reached as far north as the Bay Area, but due to human activity, have been reduced to a small area in Kern County, which consists of the remaining critical sand dune habitat in the valley. As expected, with loss of sand dune systems, we've seen insects associated with dunes also decline. One of these species is the giant flower-loving fly, and at one point in the 70s was considered extinct until its rediscovery in 1997 around the Sand Ridge Preserve. This fly occupies the only pristine sand dune within the preserve and is widely considered an indicator species of quality dunes, as its presence reflects an optimal environmental condition for undisturbed sand dune habitats. To achieve my objective of determining whether habitat restoration benefits the fly and overall insect community, 54 study plots were established within the preserve, with each plot receiving one of three possible experimental conditions. First, a control plot, which resembles the state of 70% of the preserve, dominated by invasive plants with little sand exposure. Here is where the least insect or fly activity is expected. Second, an herbicide plot, in which herbicides were used to target invasive grasses, leaving behind native shrubs and herbs. This treatment provides substantial sand exposure and native plant diversity, and I expect the most insect diversity and a fair amount of fly activity here. And third, a mowed plot, where everything except native shrubs is mowed and removed from the area. This does include the removal of native flowering herbs. The mowed treatment has the most sand exposure of the three plots, and I expect the highest fly presence here, but potentially less overall non-target insects with the removal of the native herbs. My intent is to find the treatment that best supports the flies and sustains a high insect diversity so that we can use the treatment large scale over the entire preserve, ensuring that we not only properly manage and restore the remaining sand dune habitat of the valley, but also so that we can expand the range and stabilize the population of a species on the brink of extinction. I hope that my research methods can be applied to restore other unique systems and prevent further biodiversity loss. Most notably, the Delhi Sands in Riverside County, experiencing a mirror situation with the sand dune loss and the decline of the federally endangered Delhi Sands giant flower-loving fly. Thank you.
Thank you, Mario. Great presentation. Let's give Mario a round of applause. All the participants had the opportunity to participate in a rehearsal. And when I heard this presentation and I saw the photo of the flower-loving fly, can we bring that image back up? And I thought to myself, because they thought they were extinct, there's not too many left. And I think Dr. Jackson even made a comment, but what a bummer that they look so much like a bee. Because I'm sure people think they're a bee, and some people can be crueler to bees, I guess. They want to get rid of them, or you don't want to get stung by them. And so I thought, I wonder if that contributes to the extinction of this fly. But with that said, Mario, can you share a little bit about why you became interested in your research topic?
So I work as a biologist normally, and I've been volunteering with a land management trust. And I've always wanted to be a generalist, and I normally work with small mammals, but when they told me that they had entomologists looking at a fly, I thought, can I help? And they let me help. And over the past 3 years of talking to them, I find out that entomologists in general are a dying field, and they're getting older, because aside from me, the second youngest is 70. So they said after 3 years, I'm already the next expert in line for this fly. So I figured, okay, might as well do it for my master's and find out what else more is left to learn about that, and also the preserve itself, which is, as you saw, the last of its kind in the valley.
And can you share a little bit about how you hope to contribute to the extent research and literature as it applies to your research topic?
So with this fly, it's often considered a surrogate of another fly, which I mentioned also in the sand dunes in Riverside, and that's fairly endangered, but we know almost nothing about the entire genus of this fly. But you can't do a lot of this research with that fly because it's already listed. So by me working with this fly, I can hopefully not only help this preserve that's local to me, but also help other areas that are experiencing the same situation and provide a management plan so that the federal government can actually know what the next step is to restore the habitat and restore the population of this fly. And hopefully that can go anywhere else that has sand or other systems where we can figure out, okay, maybe we should do large-scale treatments over the vegetation instead of just target the one species in general.
I think the time is now. I think you said there's only 100 left. So, well, again, thank you for your presentation. Great job. Our next participant is Jonathan Juarez, and the title of his research is Tree of Heaven Leaf Decomposition in Aquatic Ecosystems. His program is biology, and his advisor is Dr. Ray McNeish.
Freshwater habitats make up less than 1% of the total surface area of water on Earth. Despite the rarity of freshwaters, they supply water to most humans and mammals on Earth. Human activity can impact these critical ecosystems, and one way they can do that is through the introduction of invasive plant species. Invasive plant species are defined as species that are introduced outside of their native habitat and are known to have significant and negative impacts on human and environmental health. Tree leaf litter serves as a critical connection between terrestrial and freshwater ecosystems by providing energy and habitat to aquatic macroinvertebrates in these ecosystems. Common examples of aquatic macroinvertebrates include snails, dragonfly larvae, and crayfish. The invasive tree species Ailanthus altissima, also known as tree of heaven and shown here in photo 1, is common throughout the Central Valley and along the Kern River. Tree of heaven is a fast-growing plant invader that outcompetes local native species. The goal of my research is to determine how aquatic macroinvertebrates that colonize leaf litter respond to the presence of leaf litter from the invasive tree of heaven, and how this invasive species contributes to the energy available in freshwater habitats compared to native tree species. To address my research goals, I will be comparing this invasive plant to 2 California native tree species, the western sycamore, shown in photo 2, and the Fremont cottonwood, shown in photo 3. I will be using litter bags composed of mesh netting that encase leaves, like those seen in photo 4, to create a native leaf, invasive leaf, and mixed leaf litter treatments. These leaf litter bags will be anchored in 2 freshwater ecosystems. The first is at a pond on the CSUB campus, shown in photo 5, and the second will be at Tejon Creek, shown in photo 6, located on Tejon Ranch. By collecting these leaf packs through time, we can determine how fast or slow all the leaves break down and how the macroinvertebrates communities change over time. While I don't have any data to share with you because this project is still in the preparation phase, I predict that the tree of heaven leaves will break down at a faster rate than both of the native species, and they will support a different macroinvertebrate community than the 2 native species. I hope to be able to share the results of this research with you in the future. By better understanding how this common invasive species affects freshwater habitats, we can better prioritize our efforts to ensure that we maintain the health of these critical resources. Thank you.
Great job, Jonathan. Let's give Jonathan a round of applause. So first things first, Jonathan, while the judges are putting in their scores, I'm going to work to debunk whether or not number 5 is actually on campus. I'll be working on that shortly after this competition. I've not seen that area. I don't know if it's photoshopped. We were asking Jonathan, I'm over here in Numenor, and so our ponds are completely empty these days. And when he told us in rehearsals that this was actually on our campus, I said, where at? I have not seen it. So he's directed me to the area, and I promise to get back with everybody and let you know whether or not it's really on our campus. Okay, Jonathan, with that said, please share us a little bit about why you became interested in your research topic.
So I've always been a person who, growing up, we went camping a lot and we went fishing a lot. And if anybody here has ever gone fishing, they know that sitting around is 95% of it. When you're sitting around water for a long time, you start thinking about how it works and, the aspects of everything. And that's just always fascinated me. So this is right up my alley of interests.
How you plan to contribute to the extent research and literature on your research topic?
Leaf litter experiments are pretty common in aquatic ecology. But despite tree of heaven being an invasive species that's all over the country and all over the world, I only have found like 3 or 4 other papers actually studying it and how it decomposes in aquatic ecosystems. So this is going to be, hopefully be adding to that and the importance of kind of how we can handle this invasive species that's everywhere.
Thank you. Our next participant is Gracie Lyons, and the title of her research is Build My Diet Health App, Project Management Mobile Healthcare App. Her program is Healthcare Administration, and her advisor is Dr. Anthony Polito.
Imagine you have been diagnosed with a new health condition. The doctor gives you a suggested printout of what you cannot eat. You drive home, open the fridge, and think to yourself, now what can I eat? Build My Diet Health app would turn this now what situation into you knowing exactly what you can make for yourself. My research is a project management-based mobile healthcare app that will help patients create custom recipes that combines their health conditions' dietary restrictions, all with one phone swipe. Patients will be more likely to stick to their diet with an app that can do the research for them of new recipes. So how does project management relate to Build My Diet Health app? Project management plans out step-by-step a new one-time occurring project for a program, service, or product that will address an issue and solve it. My research question includes, do the 5 project management steps feasibly and efficiently develop the Build My Diet Health app? I will relate how these 5 basic steps of project management theory can be used to turn this app concept into a real-life solution. Step 1, initiation phase. This is the beginning of the project and defines what it will look like. Step 2, planning phase. This will serve as the project roadmap and will complete everything that will need to be done throughout the project. Step 3, execution phase. This is putting the plan into action and ensures that it will meet the expectations of the stakeholders. Step 4, monitoring and controlling phase. This phase mitigates risk, ensures quality, and uses a constant baseline to measure the project's success. Step 5, closing phase. This will close out the project, release the resources, ensure project payment is received, and maintain client acceptance. Client acceptance is important because it is a written, documented, and formalized way to accept that all the deliverables of the project have been done and are of quality. The initial qualitative research used are the 5 project management series steps created by the Project Management Institute. I used qualitative research, coding, and the purpose is to uncover patterns and themes in the data. These results indicated that these 5 project management steps feasibly and efficiently develop the Build My Project Health app. According to Capterra's market research report, Organizations that use project management practices waste 28 times less money than organizations that do not. These 5 phases take an initial concept, creates an action plan, and will result in a finished Build My Diet Health app. Thank you.
Great job, Gracie. Let's give Gracie a round of applause. If you could, just share a little bit about how you became interested in your research topic.
So I actually became interested in my research topic just based off experience that my family has. My family has a lot of different allergies and a lot of different medical restrictions, such as like gluten intolerance, diabetes, high blood pressure. So I came up with this app concept to solve the solution because there are a lot of apps out there where they'll address one specific health issue. But the problem with this is patients are very custom and each patient has different needs. So this way we'll combine all of the diets together and it will hopefully create a lot of stress off of them by combining all their diets together to create recipes for them. And this will hopefully not take up as much time and be time consuming for them.
In addition to the app, what do you hope to contribute to the extent literature and research as it applies to your topic?
This is the beginning research that I did before I started my thesis to make sure that it was a credible topic to choose. So what I'm going to do for as I continue on for my thesis is I'm going to actually create a project management handbook of using these 5 phases step by step, everything that will need to be completed to create this app in a real life solution. So I would be creating a budget, creating project charter, scope, just everything you can think of, ensuring a quality plan, a risk management plan. So just going through all those phases and including everything in them.
Thanks, Gracie. Move on to our next participant, Antonio Mateiro. From shrubs to weeds to shrubs again, restoring type-converted chaparral. Program is biology, and the advisor is Dr. Brandon Pratt.
So chaparral is an evergreen closed canopy shrubland that's native to the low to mid regions elevation-wise of California. It is characterized by what's called a Mediterranean type climate, or a climate that has short, mild winters that are also wet and long, hot, dry summers. And in the top left, you can see what is known as a mature chaparral. You can see all the greenery and you can't see the ground. It's much like a rainforest like that. And then the top right, you can see a type converted chaparral where the vegetation type is converted to grassland with a couple of shrubs here and there, the survivors of fire. Chaparral is impacted by fire. Now, normally in the mature chaparral, a fire occurs once every 30 to 100 years. These fires are infrequent and the chaparral can recover from that. However, due to climate change, much of the chaparral of California has been type converted into the photo you see on the top right. And this has occurred because the fires have become more frequent and more devastating, meaning that it opens up bigger gaps in the chaparral that weeds that are invasive can colonize and keep the chaparral out over a period of time. This is problematic because chaparral has 2 key ecosystem services, such as fire control. You know, the fire regime controls the chaparral, which then regulates the fire, and mudslide resistance. All those shrubs on those hillsides will anchor the soil down and prevent mudslides. So that leads us to the question of how do we restore the chaparral? Well, in the top right, which is taken at Lake Piru, I've conducted some preliminary data acquisition with my thesis advisor, Dr. Pratt, and we have generated some data on the chaparral and a related plant type, coastal sage scrub. And as you can see, the mortality data or the death rate data on Lake Piru over this last summer, April to November, shows that the chaparral have a much higher mortality rate than the coastal sage scrub. This has led me to propose that the chaparral restoration can be conducted through coastal sage scrub, which is a plant type that is much shorter than the chaparral. So the chaparral shrubs are very tall. They're like 8 feet tall. You can see on the top right photo how they sort of dwarf the grasses around them. And so I'm thinking that the coastal sage scrub can cast shade and propagate faster in the grasslands and facilitate the chaparral through nursing the plants through shade tolerance. Thank you.
Let's give Antonio a round of applause. Please share with us how you became interested in your research topic.
In my undergrad, I took a class with Dr. Pratt called Terrestrial Ecosystem Ecology. And that class talks about the different ecosystems on Earth and how they come to be the way they are. Like, why is the rainforest the way it is versus the desert the way it is? And so I really like the idea of the changing ecosystems over time, especially with the here and now of climate change. You hear headlines all the time about the rainforests in Brazil or in California. We have lots and lots of fire and in our long, hot summers.
And then how do you hope to contribute your research to the extent research and literature that's out there?
Chaparral restoration in particular is sort of in its infancy, where we know a lot about the mechanisms that are causing type conversion to happen, but we're not too sure on what we can do to restore the chaparral. So my research will provide a method that can or cannot restore it. It's part of science is that even if all my plants die, that's a great indicator that whatever it is I'm working on isn't the path to go to. So that's what it will contribute.
Well, hopefully it's successful and we're able to restore chaparral. Our next participant is Daniela Mazal. The title of her research is Pregnancy Brain is Real, and there might be something we can do about it. Her program is kinesiology, and her advisor is Dr. Zachary Zinko.
Did you know it requires the same mental load to run a family household as a Fortune 500 company? It's true. And while we're pregnant and preparing to run this Fortune 500 household, while single-handedly being judged by everyone we encounter on whether we're doing it right, our brain is going through one of the biggest changes that will occur in our lifetime. And this is for good reason. These changes are what allow us to bond with our child, to understand their needs, and to love them unconditionally. However, these same changes can have detrimental effect on something called our executive functioning skills. Parenthood is like being on an airplane when those oxygen masks actually come down. Now, we know what to do. We've heard the spiel a million times, but we freak and we put the mask on our child to suddenly realize there's no mask left for us. Now, that mask is our executive functioning skills, and we need those skills just as much as our child does. The good news is there's something fairly simple that we can do to improve these skills, and that's exercise. Exercise. Simple enough, right? Well, it sure is for the Fortune 500 CEO, but sadly in this country, not so easily accessible to pregnant people and new parents. There's extensive research that shows the benefits of exercise during pregnancy and the effects of exercise on executive functioning skills in a variety of populations. However, here in America, less than 50% of pregnant people meet the bare minimum requirements for exercise. My research aims to see, is there an effect of exercise on executive functioning skills during pregnancy and early parenthood? It's a great question, right? So how are we going to figure this out? So we're going to challenge 20 new and expecting parents to take an online Pilates class 3 times a week for 12 weeks from the comfort of their own home. And during this time, we're going to be tracking their executive functioning skills through a series of online brain games, let's call them, and compare their results to the scores of non-exercising counterparts. My goal for this research is to arm these new and expecting parents with this tool, this oxygen mask that will empower them to take on all of their wildest dreams, whether that be to be a first-class parent, a college professor, or heck, also a Fortune 500 CEO. Thank you.
Great job, Daniela. Let's give Daniela a round of applause. And Daniela, as the judges are scoring, can you just share the opening again? It's a 2-family household?
The comparison?
Yes.
Is the same mental load of running a family household to a Fortune 500 company.
Yes, and I shared that with my wife, and she said, duh. So she let me know. With that said, please share with us how you became interested in your research topic.
This is definitely near and dear to my heart. I am a mom. I'm actually pregnant with my second. I've been pregnant 5 times. I've had 3 miscarriages. So I've basically had pregnancy brain for the better part of the last decade. And I can tell you anecdotally that exercise has been very beneficial to me, but who knows if that's placebo effect or what. So I want to know if it's true, and I want to know if research supports it. So that's what I'm here to do.
Tell us how you hope to contribute to the extent research and literature on your research topic.
Like I said in my presentation, there's extensive research that shows the benefits of exercise for pregnancy, but that's actually a fairly new phenomenon. But back in the day, pregnant people weren't even encouraged to exercise. So there's very little research done on the effects of exercise during pregnancy. So I'm hoping to open up that field a little bit.
Well, great job. All right, Bella Ozuna, Let It Glow Biofluorescence in Terrica Granulosa. Programmology advisor is Dr. Amber Stokes.
Scorpions, blind squirrels, and newts. 3 animals that you would probably never group together, but that have a fascinating trait in common. So, what trait am I talking about? Well, all 3 of these animals can glow or biofluoresce under the right light conditions. Biofluorescence occurs when a living organism absorbs light and then reflects that light at a lower energy, which results in a glow that can occur in different colors. Last year, I found the rough-skinned newt Terrica granulosa biofluoresce under blue light, which I was able to photograph using a special lens filter. This research showed a pattern of biofluorescence along the limbs of the newts and concentrated on the belly of the newt. The next step in my research was aimed at figuring out the why and how the newts have biofluorescence. If newts are using biofluorescence as a communication tool between individuals of the same species, then they would most likely demonstrate an attraction to fluorescence. I tested this in the field by using minnow traps at 2 different pond locations in Oregon with glow sticks as a lure. The newts would swim in one end of the minnow trap and would be trapped until they were collected the next day. Each minnow trap would have a different colored glow stick or no glow stick, and the traps would be evenly dispersed around each pond. Unfortunately, the traps were a bust because few newts were captured. The abysmal capture rates are most likely attributed to timing. We had arrived at the ponds at the tail end of the newt breeding season, which means that there were just not a lot of newts available for capture. So, what was the next step? Well, the next step involves a tank that is basically a slightly larger kiddie pool, long sheets of aluminum, and a lot of rivets. My amazing colleagues helped me fashion together a Y maze out of the sheets of aluminum that we then placed inside the tank with water. I then attached glow sticks of different colors to the arms of the Y and released the newt into the middle of the Y facing a randomized direction. The newt then swims around the different sections, and its movements are recorded on camera for me to analyze the time the newt spent in each section and at each glow stick. I have just started analyzing these trials and cannot say much on if newts prefer one color of glow stick over another, but I will continue to test the newts for color preference to determine if this unique feature is attributed to communication between individuals of the same species. Ultimately, I hope to better understand biofluorescence because it could help us understand how newts reproduce and find mates or protect themselves from predation, which in turn may help us better understand how to protect their habitats. Thank you.
Thank you, Isabella. Great job. a little bit about how you became interested in your research area.
I started the thesis program, and Dr. Stokes, my advisor, she just gave me a bunch of different topics to look at. And one of the papers that she presented to me was published in 2020, and it was on biofluorescence in salamander species. And so we were reading through it. I found that it was very interesting. I thought it was very cool. And it wasn't until last semester when I took a conservation class with Dr. Hall that I realized that understanding how organisms communicate with one another can really just help us understand how these environments all work together. Because when you have an environment, it's not just one species alone, it's multiple species interacting, and they're they have a big impact on the environment. So if we learn how the newts communicate with one another, then maybe we can help understand how important they are to the environment that they live in, to the predators that feed on them, to the organisms that they eat. So it's all very interconnected and, very fragile. So it's important that we do research and to understand this.
Yeah, and I know you said some of your work so far did not work. But as somebody said earlier, we learned from that as well. I was impressed that you and some of your colleagues fabricated the big Y piece. And, speaking of, my grandma was a Rosie the Riveter. So when I hear anybody talking about riveting anything, I think of her. But I thought that was great, and I love the image of it. Could you share how you hope to contribute to the extent research and literature on your research topic?
Like I mentioned, that paper that I looked at was recently published in 2020, and it was talking about biofluorescence in salamander species, just talking about how these salamanders have biofluorescence, not really talking about the why, the how they have biofluorescence. And so what I'm aiming to do with my research is to lend some more clarity to that, to figure out, okay, well, are they using it to communicate with one another? Are they using it as a supplement to communicate to predators that they are toxic because the newts are toxic? Why are they using this mechanism? It's important, or else they wouldn't have it, right? So that's how my research would be contributing to the current research that exists.
Thanks, Bella. Our last participant for the evening is Natasha Selene, exploring masculinity and body image of men. Her program is counseling psychology, and her advisor is Dr. Louise Vega.
Some people think that only women can have eating disorders or body dysmorphia, but then that assumes that men don't think about themselves either. And with that information, men think about themselves in a different way than women think about themselves. And I found that there are studies that have not looked at the self-objectification theory between the male behavior of gaining muscle or losing weight. The goal of my study is to integrate the self-objectification theory with reasons behind the behaviors of gaining muscle or losing weight, and also exploring body positivity in men. So the self-objectification theory was first introduced by Fredrickson and Roberts in 1997, but in their study, they used women. The self-objectification theory states that someone looks at themselves from a third person point of view, where value is put upon them by someone else, instead of the belief of the value that they believe that they have. And in my study, I'll be using this theory to explore the behaviors of men by using a questionnaire that looks to find the correlation between attitudes and behaviors of losing weight or gaining muscles, and what influences men to be concerned or not be concerned about their body image. And I am also predicting that because men don't publicly acknowledge their low self-esteem, their beliefs are manifested in negative behaviors such as drugs, working out too much, overusing, fasting, or even smoking. And their behaviors are also affected by the influence of media, peer groups, perceived masculinity, or even their own self-esteem on whether or not they believe that they're attractive. And then I'm also predicting that the outcome will be based on whether men have autonomous motivation against body ideals and how they see themselves between their peers. In the future, this information can help therapists address eating disorders and body dysmorphia in men and getting a better idea about what influences them and what are their thoughts about themselves. And this can be a great step towards the direction of addressing the silent concern that some men endure. Thank you.
Thank you, Natasha. Let's give Natasha a round of applause. Can you share a little bit about how you became interested in this research topic?
I've had my own body image and weight loss journey, and I noticed my peers at the gym, but my peers at the gym being men, and how sometimes we would talk, like I don't normally talk to people in the gym, but sometimes people would be like, hey, because I would wear my Navy shirt or something like that. And so it came across that the guy that I was talking to was also having the same issue. He was very muscular, and he was like, man, I need to lose more weight. And I'm like looking at this dude, and he's pretty muscled. And I was like, wow, okay, so men also have this issue.
If you could just share with us, how do you hope to use your research to contribute to the extent literature and research that's out there?
Because a lot of people just assume that men just want to get buff. There's also the type of people that, so there's a lot of things actually, but the majority, it's because a lot of the research and a lot of the, when you look at manuals for eating disorders, it's all based on women, not so much based on men. And the influences that women have are different than men. And men's isn't researched as much. And there's a whole lot of things, and it's more silent than it is women. So that's where I'm hoping that with this research, I can find those silent key points that men have that can contribute to a better therapy with eating disorders for men.
Thanks, Natasha. Everyone did a great job. Hats off to the presenters as well as the advisors. With that said, I just want to remind everybody while we're compiling the final scores here, the winner will receive a trophy, much like you see there. In fact, I believe we already have it over here at the Graduate Student Center. And of course, bragging rights on campus. They're eligible for the Western Association of Graduate Schools Regional 7th Annual Grad Slam, which will happen in March in Portland, Oregon. And at that competition, they'll actually compete against competitors from the states of Arizona, Colorado, Idaho, Nevada, North Dakota, Utah, New Mexico, and several other states as well as competitors from the other CSU and UC campuses here in the state of California. Prizes at the regional Grad Slam include money. So in addition to getting to travel to Portland, Oregon, first place will receive $500, second, $250, and then there'll be a People's Choice Award. All right. So our runner-up this evening for the 2022-23 CSUB Grad Slam is Emma Barnes. Please give Emma Barnes a round of applause. Congratulations, Emma. All right. And to announce our first place winner, it is Daniela Mazal. Okay, Daniela Mazal. Congratulations. We're going to do this again next year. So for those of you that can participate again next year, please do encourage other classmates across campus to participate. And again, great job. Very well done. Let's give everybody a round of applause. Thanks again, everyone. Judges, thank you so much.